So our next speaker now is Dr. Simon Cameron, and he is Global Food Security microbiologist, and he's specialising in the study of human milk and works in Queen's University, Belfast. So thank you so much, Simon, whose talk is the microbiology of human milk.
Okay, that's great. Thank you very much for the introduction, Sarah. It's a real pleasure to be with you on this webinar this afternoon and have the opportunity to talk to you about the microbiology of human milk, some of the fascinating aspects I found out about it, give you an introduction to some of the research that brought me into this area of research initially, and some of the research projects that I have planned at Queen's University Belfast starting over the next year or so. So whenever I talk about the microbiology of human milk, I always like to start with the person who was considered the world's first pediatric infectious disease doctor. And this was a doctor called Theodore Escherich, that the bacterium Escherichia coli is named after. And he was a pediatric infectious disease doctor who worked in Germany and Austria in the latter part and the earlier part of the 19th century and the earlier part of the 20th century. And he was the first person to really link the gut microbiome to digestive health and the risk of infants dying from infectious diseases soon after birth. He was also the first person to really identify that cow's milk has a very high sodium for newborns, and it wasn't really a good method of feeding newborns in an unprocessed state, which was the current practice if a mother couldn't breastfeed for any reason. But I think most importantly, he was the first medical doctor to really emphasize the value of breastfeeding as a public health issue and launched many public health campaigns in and around Austria to really try and increase the rates of breastfeeding. And he did this because he saw that the babies who were being breastfed in his wards had a much lower risk of dying from infectious diseases that were the really driving force behind the high infant mortality rate of the day. And he noticed that these children who were being breastfed had a much lower rate, lower risk of dying of infectious diseases, particularly diarrheal diseases, than those babies who had been fed with formula. And Theodore Escherich was really about a century ahead of his time, because it's only recently in the last few decades that we've had the technology to be able to really start to understand the complexity of human milk and move away from this understanding of human milk in terms of its macronutrient components, in terms of the water, the protein, the carbohydrates, and the fats that provide the energy source for newborns, towards the much greater understanding of the whole composition of human milk, and includes some of these micronutrient components such as vitamins, minerals, important components like antibodies and immune cells, hormones, but for me, and importantly, the whole range and diversity of different types of microorganisms, including bacteria, viruses, and fungi, but also importantly, the food that helps feed those bacteria and other microorganisms called human milk oligosaccharides in the early establishment of the infant gut microbiome. So we think of this, I always like to bring it back to evolutionary biology, and this quote from an evolutionary biologist, Theodosius Dobzhansky. He said that nothing in biology makes sense except in the light of evolution. And when you think in terms of human milk, it is a high metabolic cost to the mother to produce this human milk. So every component of it must have an evolutionary purpose for being there. So this idea that bacteria and the food to feed them in human milk oligosaccharides are there, they must have a purpose in terms of the health and the advancement and the evolutionary advantage to the newborn. And in terms of the purpose of these microbes in milk and the human milk oligosaccharides and other compounds to feed them, the idea is that human milk is really fundamental to the establishment of human microbiome in early life. And we're now starting to realize and understand the importance of human microbiome to both newborn infant health, but also longer term and lifelong health as well. When we talk of the microbiome, we're talking about the 100 trillion or so different microorganisms, bacteria, viruses, fungi, protozoa that live on and within the human body. They're found on most parts of the human body, primarily in the digestive tract, but you'll also find them in the lungs, on the skin, in the urogenital tract as well. And there's very few places in the body that actually are sterile of microorganisms. There's only really the brain, the nervous system, and the circulatory system that are the key sectors where you don't want to typically find bacteria. Interestingly, we can live without the microbiome, but we don't really live very well, and you can raise mice and rats to not have any microbiome, and they will live, but they'll typically suffer from a range of different diseases. And there are a number of different diseases that have been linked to the microbiome, and these range from metabolic disorders such as diabetes to cancers to other immunological disorders. But a problem that we have with microbiome research is it's very difficult to follow different participants over a very, very long period of time. So what we normally do is we take, say, two groups of patients, say one suffering from cancer and one not suffering from cancer, and we analyze the microbiome in different parts of the body to see if there's any differences. And typically you may find differences between the microbiomes. You may find that different types of bacteria are higher or lower in abundance in people with cancer. But the problem with this approach is that it's really only correlation, and we can never really establish causation, whether or not those differences in the microbiome that we detect are what is responsible for causing that disease. So we have this real issue in the field of microbiome research, in that it's very difficult to separate correlations from causation. And I always like to come back to this figure that plots over a decade's time, from 2000 to 2009, the per capita cheese consumption in the US plotted against the number of people who die by becoming tangled in their bed sheets. And as you can see from this figure, there's about a 95% correlation between these two factors. But it's very, very unlikely that the amount of cheese that someone eats is actually a causative factor in determining whether or not they'll die from being tangled in their bed sheets. So in the field of human microbiome research, it's really important to be able to start unraveling causations from these correlations that we see. In terms of what shapes the human microbiome in early life, it's absolutely no surprise that whether or not the baby has been fed with human milk is the main determinant of what the microbiome looks like well within to the second year of life. And it's not until really from 15 months and onwards where the impact of geographical and socioeconomic factors really start to overtake the influence of breast milk, and whether that infant was fed with human milk from the beginning of their life. And in terms of when we think about what is the human microbiome doing early on in life, we think that it's actually training the gut lining in the newborn to really start to recognize friends from foe. And most microorganisms really don't want to cause diseases in humans. They really are just the wrong bacteria in the wrong place at the wrong time in the ones that cause disease. So actually the ability for the newborn's immune system to be able to detect friendly bacteria from aggressive bacteria is really important. And the presence of these microorganisms in human milk and these human oligosaccharides that help to establish the human microbiome from early life are really important in training this gut lining to not really effectively overreact when we see other microorganisms. So when a baby isn't being fed with human milk, what we think is happening is that this symbiosis, this relationship, this kind of balancing act between the microbiome and the infant's gut lining isn't established in a balanced way early on in life. So the infant's immune system may overreact or act incorrectly when it observes healthy bacteria later on in life, and causes a lot of issues, particularly around inflammation, which could go on to contribute to different metabolic disorders later on in life. So human milk contains microorganisms, as I said, but for quite a long time we did consider that human milk was actually a sterile fluid, that bacteria and other microorganisms shouldn't actually be in there if it had been processed aseptically. But we now know that human milk contains well over several different species of bacteria and other microorganisms, but we're still not really sure when they come from. This is quite interesting because typically adding microorganisms to food is generally considered a bad idea in essence for defined purposes such as cheese production. So as I said, we don't really know where these microorganisms come from, but it's probably a combination of different routes. What we do know is that the first feeding of a newborn, that milk does contain microorganisms. So it's not likely to have come directly from the infant through a sort of a mixing of the oral microbiome to the microbiome that's found in human milk. It's probably associated with this, but also physiological changes during pregnancy, where the growing fetus causes sort of a rearrangement of the gastrointestinal tract that moves it closer towards the breast tissue, and the permeability of the gut membrane changes, which allows this translocation, this movement of gut microorganisms into the breast tissue, which can then go on to establish gut microbiome in newborn infants. But really interesting, as I mentioned earlier, human milk not only contains the microorganisms to establish into the newborn gut microbiome, but it also contains the food to feed them. And this is one of the most stark differences between human milk and bovine milk that's used to make formula. And this is in the concentration and diversity of different human milk oligosaccharides. And these are a type of sugar that are effectively an extension of lactose, and they're a large energy source that's not typically or not consumed by the baby. So bringing it back to an evolutionary perspective, this means that there has to have been a purpose for these human milk oligosaccharides to be present. And they're very different to bovine milk oligosaccharides that you would find in cow's milk. So because of their size, their concentration and diversity, it seems to be that evolution has evolved to have these human milk oligosaccharides in human milk to really drive and shape the human gut microbiome in early life. So what brought me into human milk research was actually a citizen science project I worked with Dr. Natalie Schenker when we were both at Imperial College in London and a parenting science camp. So what we were interested in this study and working with about 120 breastfeeding mothers who all came to London to donate breast milk samples were what's actually in breast milk and human milk composition up to 48 months of the nursling age. So we were looking at milk that was being used to feed nurslings from 3 months old up until 48 months old. So what we did with this is we analysed it with a type of technology called mass spectrometry that gives you an idea of the different chemical composition of human milk, and this is an area that I work in, and I'm hoping a video may work. On the right hand side, this just shows how the it looks like it's not going to work, unfortunately, today. But basically what we do with the milk is we heat it up really, really quickly with a carbon dioxide laser, and then we analyse the chemical constituents of the smoke that's produced through this heating with mass spectrometry, and it gives us an idea of the different chemical components of the milk. So what we found when we looked at the macronutrient features of human milk, looking at how much volume was expressed, the fat concentration and the microbial load of the different types of milk, is actually the only differences between the different age groups was that the volume that was expressed by the older age groups above 24 months of nursing age was really the only difference that we saw in the expressed volume reduced after 24 months, and this is probably because of the frequency of feeding. Interestingly, we didn't see any differences in terms of how that milk was expressed, if it was hand or using manual or electric pump. But we also didn't find any differences in microbial load, which is particularly important because there isn't any concentration of bacteria that could potentially be harmful to babies. But when we're looking at the chemical constituents of human milk, we can use different statistical methods. On the right hand side, this is an analysis of what we call a principal component analysis, and basically the message here is that the closer the dots are to one another, the more similar they are to samples or other dots that they find here. So when you can see these different colours overlapping, it tells us that there's no difference between the milk as we can detect using this type of analysis. And here you can see that there's no significant difference between expression methods that we used. But what we're particularly interested in was whether or not there was any difference between human milk across different ages, but we also compared it to different types of formula milk. And this is what I find really fascinating about human milk, is because all of the milk plots all sit very, very closely together in the chart. And that means that there's really no difference in terms of the chemical components that we can detect using mass spectrometry. And these were formula milks that were sold to feed babies of different ages, so from 0 to 6 months, and then 6 to 24 months, 6 to 12 months, sorry, and 12 months and above. But on the human milk, on the breast milk plots, there's a huge diversity in the different types of chemicals and different concentrations of each that we can see. So this shows that there's a lot of diversity within human milk samples coming from different mothers. But importantly, we didn't see any significant differences between nursling ages. We also looked at the microbiome that's found within human milk, and we saw no differences between the different age groups of nursing ages, and we saw no differences either between how the milk was expressed, if it was using hand or an electric or manual pump, which is really important. But we also, really interestingly, found a lot of correlations between the human milk microbiome, the bacteria that were found in the human milk, and other components that we could detect using mass spectrometry. And a lot of these components aren't actually human milk oligosaccharides. So for me, it suggests that the complexity of human milk is even greater than we originally thought, and it's not just human milk oligosaccharides that are important for establishing the human microbiome in newborns, and this is an area of research that we're pursuing even more now at Queen's University Belfast with Natalie. But what the issue we have is with this type of analysis is when we sequence bacteria using a specific gene, we're only able to achieve taxonomy down to the genus level. So this is a little bit similar to going just to, we aren't able to go to the species level. So when we look at this in terms of the animal kingdom, with bacterial sequencing, we're only able to go to the equivalent of the Equus genus in animals. And in the Equus genus, you have donkeys, you have zebras, you have horses. But in our sequencing method, all of these would be considered the same. And we don't really get the idea of the different species, how they're interacting, what they look like, how they live. And this is a real limitation of microbiome research at the moment. But understanding this is going to be really, really important in future work, and it's what we're pursuing at Queen's. The problem as well is a lot of microbiome research only looks at bacteria, but we know the microbiome is more complex than that. It contains fungi, it contains viruses, and it contains archaea as well, and other microorganisms even more. And these all interact and talk to one another and interact with the host as well. So we're actually, with our microbiome research, only looking at a fraction of the entire human microbiome at the moment. So for me, this is really exciting. It means we've got a really interesting career of research ahead of us. But it again tells us that the story that we're seeing is only a slight picture of what's actually happening. So to give a very brief introduction and to end with some of the projects that are beginning at Queen's University Belfast over the next year or so, as hopefully COVID restrictions die down, is that as a community, microbiologists are really bad at growing a lot of microorganisms from a range of samples. We think we can only grow between 1% and 5% of microorganisms from a sample. And very little work has actually been dedicated to trying to improve the culture of microorganisms from human milk, and we need these cultures to be able to study them biologically. So this research at Queen's is going to be looking at different ways of simulating the human milk composition to be able to really maximize the amount of different microorganisms we can grow from human milk to be able to study them in the lab. We also want to try and model how microorganisms grow in the infant gut. And this is because sampling babies can be particularly difficult, and interacting with the gut microbiome in babies is obviously challenging ethically and practically. So we're going to try and replicate what the baby's gut is like inside the lab so we can model it against different microorganisms, such as those involved with necrotizing enterocolitis that Natalie talked about earlier. We're also particularly interested in whether or not microorganisms grow best in a milk that they're from, or whether or not they can grow equally as well in milk from another mother's milk. And this gives us an idea of whether or not the human milk is specific to the microorganisms found within that specific sample of milk, or if there's more general ideas of how milk is supporting the bacteria and other microorganisms that's grown and found within them. We're also particularly interested in looking at how storage affects the microorganisms in milk, how it's frozen, how it's stored, how it's expressed, because this is particularly important in terms of safety of milk, but also in ensuring that the beneficial microorganisms are passed on to the newborn. But we're also really interested in finding cheaper and quicker ways to profile and phenotype milk. The type of mass spectrometry that I talked about earlier is very expensive. It costs anywhere up to about £500,000 for a piece of equipment to analyse these human milk samples, which is obviously well beyond what many human milk banks and many hospitals and other organisations is able to afford. But this is really kind of taking a sledgehammer to a nail, but we can access other smaller pieces of analytical equipment that are the size and price of typical desktop computers or mobile phones. And what we're interested in doing is seeing if we can answer questions such as what's in the milk, is the milk safe, and is there best use for the milk? So could it be targeted towards babies who are suffering or at risk of necrotizing enterocolitis? And this could be done in the milk bank or in the neonatal intensive care unit using very cheap, easy, and accessible equipment. So that, I'd like to apologise for going a little bit over time, but also really try and reinforce this idea that human milk is far, far more complex than we ever thought about, even just a decade ago. And we need a wide range of experimental approaches to really ensure that we can start to discover the links between the components, composition of human milk, and establishing these causative relationships between early life feeding and the microbiome and lifelong health. So thank you very much, and more than happy to answer any questions.